The rain came quickly. Afterwards, moths swarmed the algae-covered lamp above the terrace. In the animated movie Princess Mononoke, plants glow in the footprints of the nature spirit. One breath later, they wither and dissolve. The sound of water drops striking the leaves. Entropy. So the rain is transformed into a distant roaring. And the pale blue dot is a photograph of planet Earth, taken in 1990 by the Voyager 1 spacecraft from a record distance of about 6 billion kilometers from Earth. October 10, 2011, a 24-hour-long clip of the engine sound from the fictional starship USS Enterprise was uploaded to YouTube. The full designation of the ship is USS Enterprise, NCC 1701D. By May 25, 2015, the clip had been viewed over 2 million times. Safety, well-lit rooms, someone always awake, a heart beats, and one exhalation later. The illuminated trail, 8:30 p.m. Low cloud cover, no stars, high levels of oxygen. The few intact light bulbs dimmed by thousands of water drops. Sections passing through the swamp flooded. The water might be waist deep with the next step. The number of exoplanets in the Milky Way is estimated at 400 billion. Approximately 17 billion of these have Earth-like characteristics. When Ursula K. Le Guin, in The Farthest Shore, describes the distance between islands, does she mean life, a silent green explosion in space, unpredictable glow, complex information, a leaf in the wind, in the darkness? Passing through the islands of light, the shadow of the insect in front of the window, the outline of the planet in front of the sun. Pulled up the hood, forced myself not to remove the headphones, looked over my shoulder several times. The lamps swaying in the wind, leading back into the trees. Over the course of several years, the asparagus plot transforms. covered by weeds, turning into meadow, into brushwood, the beginnings of a forest. The slow rush of vegetation up towards the garage. So the clouds came closer to the ground. And if something were to approach, the panic lights above the lawn leak into the fields and trees. Every animal stops in its tracks, eyes reflecting light. And inside the house, mechanisms are activated, window guards lowered. I go down into the basement, through corridors, steel doors slamming behind me, a system of airlocks. All the way back to the storeroom, into the crawl space. Wait. Watch the screens. 15 minutes, an hour. Then the locks start to give way one by one. The sound of steel bending. Shadows around the corner, preceded by tiny particles of light. By a smell of freshly cut grass, rotting kelp, fir tree forest warmed by sunlight, and that undertone of smoke and turpentine. Maybe it can be studied the way you study a previously unknown species. Something moves restlessly in a glass cage, swinging its tail. Light reflected in a pair of eyes. The constants shift. Return to the shadows. Formulating a poetics, accessing the fundamental structure. Crystalline formations, slides of rock types or plants. The syntax resembles poetry. It moves in unpredictable ways, with a set of rules too complex or decentralized to take in. Or a novel about rooftop gardens. It's raining, of course. Tall grass, a flock of birds taking off. The only intact lights lit during daytime. The road covered in moss, a slow soundtrack. The pizzeria in the park, lost in a wall of forest, steaming, whistling, squeaking. And images moving gently. Old leaves remain until spring. Eat canned mushrooms. Watch condensation form on the outside of the windows. Time. The only lights, the emergency generator of the hospital. The car is still sinking. The electricity quietly wandering in the treetops. And it's raining, of course. And I wrote, ask, and the laws of physics are repealed. the wind carries red sand, is it visible? Starry sky scattered within the branches of the pistachio tree until we turn away. And then it continues. You can't look straight at it. Ask the hum of branches ringing in the body, a nervous shimmer, change inside the frequency, therein a tone, blood red. Listen quietly to the storm until we turn away. The pattern of the wind twisting, a theory of everything, a rush of heat to the face. Ask. Gravity, radiation, making it visible. To accept that is music. Notes from a meeting. Giving is central. Wanting to ask, not answer. And the universe expands. We thought we could control the night. And it continues. Thank you.
Thank you, Johannes, for that beautiful performance. This was Johannes Heldén with Astroecology. And you couldn't see it because it was in back of you, but as you said the word moth, there was a tiny moth flying on the top of the screen, so that was a beautiful moment. This is the last event of School of Non-Humans, and the idea of school involves training, learning, pedagogy, but in a broader sense, it also involves schools of thought, ideas shared by large groups of people. So I hope that after tonight's triple bill, you will come away with a sense of the possibility of creating new ideas shared by a large group of people, mainly you. The idea that it is possible to create a better future where we are kinder and gentler to the other species that we share this planet with. Tonight is also the feast of the Day of the Dead in Mexico, so perhaps it is quite fitting that we chose to name this event the Night of the Non-Humans. I give you over to Pauline Drescher, who will introduce tonight's speakers. She's a curator and researcher. Thank you, Pauline.
Thank you, Barbara. Well, welcome everybody. We are having quite a night ahead of us. We will start with a keynote presentation by Kathy. After that, there will be a surprise break with some drinks. After that, we continue with 3 more presentations and a panel discussions. So let's start with the first presenter, Kathy. I put on my glasses. Kathy Hai is an interdisciplinary artist working with technology, art, and biology. She collaborates with scientists and artists and considers living systems, empathy, animal sentience, and the social, political, and ethical dilemmas of biotechnology and surrounding industries. She has received awards, including from the Guggenheim Foundation, National Endowment for Arts. Hai is a professor in arts and has a lab at the Center for Biotechnology and Interdisciplinary Studies, Rensselaer Polytechnic Institute in Troy. Her artwork has been shown at the Museum of Modern Art, the Science Gallery, NGBK, and Maas Mokka. And she has residencies with Symbiotica and Finnish Society of Bioart, Coalesce University of Buffalo, Girassi Scientific Delirium Madness, and at the moment she is working at the Palo Lab, the University of Seattle. I think for now we give you the floor, Kathy. Good luck, and we're looking forward to your story.
Thank you so much for that introduction, Pauline, and thank you to the curators for this lovely School of Non-Humans, which is quite impressive. Okay, so I am going to speak about my work, but also try to weave a story for you in terms of how I have been thinking about working with other than humans, and of course humans too. So I'll try to get into that, and please enjoy. So I've had a background as a filmmaker for quite a number of years, and early on was doing films about women as patients dealing with the medical system, and so how bodies have been experimented on and looked at is a subject that I've been very interested in. This is an early film of mine from the 90s, which was about new reproductive technologies, which actually haven't changed that much, surprisingly, since the 90s. In the late 90s, early 2000s, I was working on a project with animals, specifically with interspecies telepathy. So you've probably heard of animal communicators and people who actually speak with animals. And so this was something that I became very intrigued with, and it started to shift my way of thinking about animals because I had different modes of approaching them and communicating. Of late, I've been working with these thoughts about the so-called Anthropocene, which many of us have been, and have been working with a collaborator whose name is Oliver Kellhammer. We have been working on films and other projects. This is a project that I've been thinking about. I've bought a gravesite, which is a natural burial gravesite, which I don't know if it's as common here, but they're becoming more common in the US. This gravesite is, set in the country, and they've given it complete instructions as to what kind of gardens can be held above the bodies. So I've been doing designs with bodies of animals that I've found, since it's not really legal for me to be burying humans. So to try and test out what helps these plants of mine, and indigenous plants specifically, grow. Oliver is, as I said, my collaborator, is also a permaculturalist. So he's been working for a number of years thinking about landscapes. And one of the things that we've become very interested in is ruderal landscapes and ruderal ecologies. This is a term that has become more in use with urban planners of late, thinking through what grows from the ruins, what grows from the pavements, what are plants that are so resilient and adaptable that they can do that. So we've been looking at these. And Oliver asked this amazing question at one point when we were filming, and he asked, what are the correlations between what is happening in our bodies and in our guts and on our skin to the microbiological processes occurring in urban air, soil, and waterways? So how are these connected? Are there novel symbiotic partnerships for bioremediation and detoxification evolving under our very nose? So we're both looking at these questions, and I'll follow this through as a theme later in the talk. I became interested in bioart, particularly in this field, which has been taken off maybe in the last 20 years, although it's been with us forever, but in terms of contemporary art. So this is about manipulating life and the art that comes from that manipulation. And I'm never certain how much people know about this field, so I'm giving a teeny bit of background on it. Some of you may be familiar with this image that occurred in the late 90s from two scientists, the Vacanti brothers. It was an ear on the back of a mouse, which was actually talking about synthetic organs, but it was really just a scaffolding underneath a mouse's skin. But it became, in the 90s, a polarizing image, horrific to people, about this kind of future that we might have. And of course now it's kind of ubiquitous. We see these images all the time, and they're kind of commonplace. An artist who I quite respect, Marta de Menezes, who's a Portuguese artist, made a piece in the late 90s and 2000 called Nature, ? Where she was beginning to look at this divide between nature and culture, so-called divide. She took butterflies and actually started to augment the designs on their wings. So you see these little patterns on there. She introduced a hot needle into the cocoons of the butterflies where they have no nerve endings, so they felt no pain, and began to augment, as I said, their design. And she asked this question then, what does this mean? Is this something coming on? Is this a design by the butterfly itself, since it's actually manifesting it, or is it something that's man-made per se? And where does that line is drawn? A lot of this work begins to ask these questions. Brandon Ballingy is a scientist, a biologist, and an artist from the US, and he's been working with amphibians for quite a long time. This is an image where he scanned the cartilage and the skeletal structure of this tree frog to try and show us the kind of deformations that this frog has. The deformations are caused by toxins in the environment. So Brandon is actually also an activist, and this is something that I admire quite a bit about his work. So I think that one of the things about all of these pieces that I'm showing you is that kind of ethical questions that people begin to, and cultural questions that people begin to bring up are really of deep importance to me, and maybe the reason I started to move into this area. Tissue Culture and Art is a group that works in Symbiotica in Western Australia, and it's made up of Yonette Zur and Oren Katz, and they work with tissue culturing to create sculptures. And this is perhaps one of their most infamous sculptures called Victimless Leather, which was an idea to possibly create leather jackets without using leather, without using animal products, and it has to be presented, though, in this bioreactor. So, and we're not sure where it's going to go. They ask these questions, but the other thing Oren and Yonette always talk about with this work is it's not really that, animal cruelty free, because there's a bovine serum that has to be used to feed the cells in the tissue that are making up the little jacket here, and they're coming from calves. They're coming from calves' blood. So nothing is really without cost to other creatures, it seems, at this point, and we're all trying to figure this out. This is an early piece of mine that was working with lab rats called Embracing Animal. And I was particularly interested in working with transgenic rats, rats that had been infused with human DNA to make them closer to human diseased conditions. So they're created in the lab and they're used for pharmaceutical research. And I created a living habitat for them in one of the museums that they were shown in, Mass Mocha. And this habitat was like a rat paradise. It was, like 7 meters high in the back or in length and about 3 meters high. And they had hiding places and all of this. And I was interested in this because I have Crohn's disease and other autoimmune diseases. And I realized that these rats had been developed to create medicines to treat diseases like my own, and it was a kind of reverse engineering. I was like curious, what were these animals? How were they created, and how could we intervene and maybe sustain their lives a little bit in a different way than they might have in the lab? So I was influenced also by this quote from Donna Haraway, and Oncomouse, which is a cancer breeding mouse. Oncomouse TM is my sibling, she says, and more properly, male or female, he is my sister. And her essence is to be a mammal, a bearer by definition of the mammary glands, and a site for the operation of a transplanted human tumor-producing gene, an oncogene that reliably produces breast cancer. Above all, OncoTM, mouse TM, is the first patented animal in the world. By definition, then, in the practices of materialized refiguration, he is an invention. I took on these rats because I really was curious how to treat them, as I had been with my own diseases. I had been working with alternative medicines quite a bit, treated these rats with homeopathy and lots of other different kinds of care than they had had in their past. At the end of it, they lived longer than they should have. I had interviewed one of the researchers who had developed them, and we were quite happy that this had turned out, because I kind of hadn't known. It was a bit of an experiment on my part. And I took them home after the long 10-month exhibition, And rats don't live very long. When they're healthy, they live only about 3 years. And these rats had about 1.5 years expectancy, but they'd already lived that long. And I thought, well, they maybe have a little bit of time left. And after I got them home, the night watchman came to me, who had been on duty at the university, at the museum, and said, can I take them to my house? Because he'd fallen in love with them. We hadn't changed their circadian rhythm, so they'd been playful at night when he was on duty, and he really wanted to be with them. So that's what happened with them. They went home with him and his wife, and they died with him. I was lucky enough to go to Symbiotica, which is a science and art residency in Western Australia, as I said, and there I continued some of the study, which I called under the umbrella of Vampire Study Group, where I continued thinking about liminal space and liminal animals. Crafted through the ordinary practices that make metaphor into material fact, Onkomouse's status as an invention, which remains a living animal, is what makes her a vampire, subsiding in the realms of the undead. This is another continuation of that quote from Donna Haraway. The undead, animals and cells and whatever that can't be sustained except in the laboratory. So I again was working with rats. And I was working with rats in the lab, and I wasn't able to photograph them, but I could do kind of funny sketch drawings of them when they came to greet us in the morning. And I realized at this point in time that rats had a whole register of communication that I had not understood before, which was also in the ultrasonic range, way above what humans can hear. We can hear up to about 20 kilohertz, and rats can hear from there until over 100 kilohertz. And I was fascinated with this because I was interested also in what was their lab experience like, if they're hearing this whole other range of things that we can't hear. And I took a bat detector, which downsamples these ultrasonic sounds, into a laboratory and started listening to just the typical practices of treating the rats, and realized that they had, there was some really crappy sound in there, like things just went off in my headphones. I was like, what is that? I turned to the attendants, and they just had taken plastic off a syringe. It was a very common practice, but it actually probably was really disturbing and stressful for the rats. So they started to change some of the practices of the lab. So that was great. But then I learned this other thing, which actually kind of blew my mind. So I'm just going to show you this video that I had found when I was there.
As we have listened to animals playing, we have heard what appeared to be the sounds of laughter. And we studied these for a couple of years without quite understanding that this might be laughter. And then one day we decided to tickle some animals. And we realized that we had to look at the sounds at a very different register than we can hear. So we obtained these transducers that are called bat detectors, that can bring very high frequencies down to our auditory range. And when we did this and we listened in, we could tickle animals and generate a lot of vocal activity that appeared to be laughter. These animals would begin to enjoy our company, and they would start to play with our hands, and wherever we would put our hands, they would follow it. And when we tested these animals to ask whether they were enjoying this kind of activity, the unambiguous answer was yes.
So when I had been working in the laboratory, I noticed when we left at the end of the day, the lab attendants were trying to tend to the rats by leaving the radio on, and they would often put on these bad country western stations or, rock music, like heavy metal. And I thought, I wonder if that's really what they wanted to hear. And I started at this point, and this was literally 10 years ago, to think about making them a chorus of rat laughter. And this is the piece that I have in the exhibit. And it took us literally that long because it's very hard to work with this ultrasonic material as an artist because A, the equipment's very expensive, but then also getting your hands on the material. And that guy, Jake Panksepp, was really one of the leaders in the field around this, but he never answered my emails. And so I finally wrote to one of his collaborators, it dawned on me. And so Jeffrey Bergdorf, who very kindly sent me reams of material this summer. And we started really working with the files. So a lot of this you can hear at the exhibition, but I'll just play you some of this, which is downsampled for us to hear. And it doesn't sound like much. It's kind of the aesthetics of disappointment in some way, but it is quite amazing that what we're listening to is a kind of layering of different rats laughing together, which is artificially edited, of course, but we're hoping when we go back home after this exhibition with the equipment, we can actually introduce this to the lab rats at my university and start working with them to hopefully lighten their load a bit for a moment. I don't know. I had many collaborators on the project, so here they're all listed. They were quite incredible, including one of my PhD students, Michelle Temple, and the technologist Matt Wellens, as well as Jeffrey Burgeth and others. And so please go look at this project. It raised as many questions about what we don't know in terms of this kind of communication as it did what we do know. And really, I think we're scratching the surface of any of this communication with animals. I'm just briefly going to go into, oh wait, it's starting this thing again. Some projects that I did leading into me working with cells. So this was again at Symbiotica. It was a project called Blood Wars, where I took blood samples from 2 different competitors and started to put their white blood cells together to see what would happen. This was a project where I was really interested in the language around the immune system that we use, this language of aggressive white blood cells fighting off the intruders. And I modeled it after the World Cup tournament. So you might know of that. You take 2 matches and whoever the winner is play each other. And so this can be upscaled as much as you want. We had really fierce competitors, and people actually really came to me saying, please, can I give you my blood? Like lots of people. And so I developed this process where I learned how to separate the white blood cells out from the rest of the material and stained them 2 different colors and put them in a Petri dish together, so the 2 different people's white blood cells. And then they were put under a microscope, and with time-lapse microscopy, we could follow them for 8 to 10 hours. And I expected them to just, have this battle, but they didn't. They did sometimes. I'm just going to show you these films of the cell wars as we're looking at it. Look at a small portion of the screen so you can see the kinds of exchanges. But I worked with an immunologist who was helping me interpret them, and he said he was kind of intrigued with this because they weren't just fighting. They were sometimes making love. They were sometimes deciding to mutually die, like a Romeo and Juliet scenario. And so the thing that this taught me was just how much I don't know and we don't know about the immune system. And that is always a great challenge for an artist or for any of us. I then became very intrigued with the research that was being done with the gut microbiome. Again, as you know, I said I had Crohn's disease, which is an inflammatory bowel disease. And I thought this whole research area was just absolutely fascinating. And of course, having Crohn's disease, you poop all the time. And so you're just like, it's something I was really ashamed of. And then I realized shame is part of the issue around this whole area. And I'm not making this connection very well, but this film I saw, Miranda July's Me, You, and Everyone We Know, which is a really wonderful film if you haven't seen it. And there's this incredible moment in the film where these 2 young boys get on a chat line and they're trying to seduce this woman, and they're literally like 10 and 6. And the older brother says to the younger one, what should I say? And the younger one says, say this. You poop into my butthole and I poop into your butthole, back and forth forever. And they did, and the woman writes back and says, that's really hot. So I realized, okay, poop is shameful, it's also hot. What is this? So there's a whole area that I haven't unpacked yet. I'm just going to leave that with you. You can tell me later what you think. I sent my poop away to get analyzed, like we do everything these days. And so I realized that I have skewed stuff there in my microbiome. And that was not that surprising to me, but it kind of set me going, well, what does that mean? What is that about? And how can I figure this out and follow it up? And there was follow-up emails. I've sort of put this together to try and save the room, but things like, are you depressed and anxious? Oh, that you could be depressed because of your microbiome, or you could be getting some, that could be the reason you have other diseases is because of your microbiome. And I was like, wait. And this was, 5 years ago or so. There are also all these products now on the market. I don't know if you have them here, but Mother Dirt is actually a bacteria from soil that is marketed to replace soap because we're really too clean. We're way too clean. And so this is a great product. I've tried it. I haven't been able to do the whole regime, but maybe you can. And then I became obsessed with fecal microbial transplantation. And some of you have seen, maybe we're sat in on the film today, so forgive me for being redundant with this. But fecal microbial transplantation is taking poop from one person and putting it in another to treat them. And this is incredible. And so I worked with a couple of collaborators, Guy Schaefer and Alison Burkoy, to develop this project, where we went around and interviewed scientists in the field who were doing this research. Because at the time when we started, about 5 years ago, we didn't know that much about it. And so we wanted to figure it out. So I'm just going to show you a couple minutes from the beginning of it so you get a sense of what that's like.
So FMT is fecal microbiota transplantation, and what we do is we take a donor stool and we transplant that stool into the recipient's body. The whole goal is to, in essence, we are transplanting stool, but the goal is to give the new microorganisms, the bacteria, and to have a chance for the new microorganisms to actually implant into the recipient's body. And we do that either through a nasogastric tube, where we make a solution of the stool. We do a colonoscopy and then take the donor stool's solution and spray it into the recipient's intestines. We can also offer it through enemas or through flexible sigmoidoscopy, which is like a mini version of colonoscopy.
Open Biome actually started in part out of some of the research that I was doing at MIT at the time, investigating the human microbiome, and then in part also out of a family friend who ended up getting Clostridium difficile infection. He ended up going on 7 rounds of vancomycin, which is sort of the most powerful antibiotic we have for C. diff. Sort of failed every antibiotic treatment that he was on, and was very, very desperate for an alternative, something that would work for him. He ended up doing fecal transplant at home in his apartment with his roommate's stool, and it worked for him.
Okay. So, oh wait, is it trying it again? Sorry. Keeps doing that. So this became, I became obsessed with this notion of fecal transplants. And a good friend of mine, who's a performance artist, Kira O'Reilly, said to me, you know, so if you could have a fecal transplant from anyone, who would it be? And of course I said David Bowie. So, I mean, wouldn't you? So this was before he died, and I produced a series of photographs where I was emulating some of my favorite images of his. He'd been quite inspirational to me, especially in the 70s and 80s. These photographs are made by my dear friend Eleanor Goldsmith, and I then sent them to him and sent him a letter that said, "Dear David Bowie, I have a bargain for you. I'm writing you with a strange request. I've been a lifelong fan. I've been following your career since I was little. You know, I was born in 1954, so not that much younger than you, but enough to feel like a younger sister. I offer these photos to you, reenacting your famous images of your career. I know thousands of fans have done the same, Tilda probably the best, but I humbly offer mine among the others. I was hoping these photos might capture your attention for a moment, and I want to exchange these for a throwaway item, your poo. I want to conduct a fecal transplantation with your stool implanting your poop into gut biome into my colon." So I was convinced that this would give me a little of that Bowie sparkle, you know, and be more like an organ transplant than just like a poop transplant. You know, it really give me something special. And, you know, sadly, I didn't know. He was sick and about to die, and working on many, many, many projects, which he thankfully got through before he died. Hopefully, he probably had many others. But anyway, he didn't answer, and so that makes good sense to me now. Since that time, I've also been involved with a lab, as Pauline said, with the DiPaolo lab, which has been amazing. I approached this scientist and asked him, he works with gut microbial research, and I said, "Can I be embedded in your lab?" And he was like, "I don't know. I've never had an artist come on over." So I spent quite a bit of time with him and his people, and they're fantastic. And we started working with our own poop, and we did a couple projects. The first one being Will's was the healthy poop, and mine was the unhealthy poop. We developed a project where we took poop samples, as I said, and blood samples, and kind of introduced blood samples to the poop samples. This is a design that Will was interested in. He designed this project completely because he thought that because of the way my gut works, I might have already had my blood introduced to my stool from the Crohn's. Well, he possibly was right, but when we started following the research with his poop samples introduced to both of our blood, it looked kind of the same. Nothing was really happening. And then we introduced my poop samples to the blood samples, the white blood cells, and his are on the bottom and mine are on the top. And I'm like, what is that? And that turned out to be my T cells going crazy over this introduction of my own bodily material, my gut biomaterial. And so this is a typical autoimmune reaction, you could say. Oh, it's overreacting. But at first I thought that, and I was kind of horrified. And then I realized this is a beautifully poetic image. And maybe my body is actually a mutant. Maybe I'm actually ahead of the curve instead of behind it or disabled, as it's known, a disability. And maybe it's something that we're going to have to think about changing towards. So I'll talk about that more in a little bit. Our research developed into some other things, the family biocrests, where we're looking at what microbiome people share, people who live together might share. So I made these family crests of their bacteria that they share. So we collected lots of poop samples from people and their pets, plated them on various kinds of agar, which expressed different kinds of bacteria. I'm really happy about all of this, you can see. We analyzed the samples in 16S, which is a DNA way of analyzing them, and then also proteomics, so that we could see things that were close to each other. All of these people and their poop are tagged in numbers, so you can't see who they are, and nobody's identification can be given away. But that's how I was able to determine these kinds of groupings of shared, like everybody has E. coli, so that's going to be a given. But Klebsiella pneumonia, for example, or Mycococcus luteus, this family of 3 adult women, 1 baby infant, and 1 feline cat. This is what they started to share. In this family of 2 adult women, they share different bacteria, Enterococcus faecalis and Bacillus subtilis, as well as the E. coli. And this goes on and on. But while we were doing this research, something else happened, and this is one of the plates of Will's and my bacteria, fecal bacteria, plated, and kind of we're looking at what the different bacteria is on this one plate through bacterial morphology, which is a kind of way of identification just by shape. And I started seeing these blank areas in these plates. And here was another one where I was like, wait, Will is expressing, his bacteria is expressing really well, and mine is absent. And this happened again and again and again. So this observation of absence and this what I called landscape of lost microbes was really kind of disturbing to me, because this means that I have a very state of dysbiosis. There's not much diversity of bacteria in my gut. And I thought, what can we do about that? Being a problem solver. And we always want to think about our gut as a hackable space. We'll take probiotics and all of this. But again, my collaborator, Guy Schaefer, and I talked about it, and we thought, well, maybe we could extend the research that we've been doing with fecal transplants and think about a different DIY way to do it. So we started thinking about these kind of new project, which is taken off of OkCupid, the dating service called OkPoopid, where you could possibly meet your perfect mate, poop match, through this kind of interview process. So it's kind of, we've only developed the video promo, not the app yet, but I will just play a little bit of it for you.
Oh, hi.
Hi. It's so good to meet you.
It's really good to meet you.
Yeah.
Thank you so much for
Of course.
coming.
Yeah,
This is
I really
Guy,
want
by
to be
the
able
way,
to
one of my collaborators.
help out with this.
Oh, that's really, really great. It's awesome. You're the first, so I'm a little nervous.
Thank you. I'm really honored.
Yeah. Yeah, me too.
Yeah.
No,
Oh, of course, yeah.
like, and I wanted you to be first, so, you know.
It's really touching. I mean, it means a lot to me that you thought that. So thank you.
Yeah, so maybe we should, I mean, maybe we should just get down to business and I'll ask you some questions.
Yeah. And feel free to ask questions. I'm
Oh.
an open book with regards to my poop.
Oh, God, thanks.
You're welcome.
So, like, what do you like to eat? I mean, are you vegan?
Oh, not at all. I eat everything. I mean, I'm a fairly, there's a lot of salads, a lot of
Yeah.
oatmeal, a lot of whole grains, a lot of, roasted vegetables. But there's meat. Any meat. Yeah.
Meat, okay.
Fish, yeah.
Meat, okay, yeah. And how's that work out for you? I mean,
The meat, I'm a little sensitive with regards to meat.
Yeah.
I had kind of a big pile of meat last night, and it didn't. There were problems, I mean, sometimes I overload the meat.
oh, okay.
Yeah, sometimes.
And
And it just kind of backs up a little bit, and it hurts, and yeah.
What sign are you?
I'm a Scorpio.
Oh, me too.
Oh my God. Oh my God, that's so
we
funny.
might really be compatible. That's
Oh
amazing.
yeah. Of course, Scorpio poop. So yeah. That makes sense.
Wow, that's so amazing.
So yeah, this is really funny and kind of absurd and whatever. But I mean I do think about this kind of seriously because I come from a privileged position. I'm a white woman in the U.S. and I have a teaching position and and so I'm situated in middle class squarely, and whatever that means these days. And, of course, to work on my gut, I think about things like, I should be eating, an organic diet, a vegan diet, and, watch the meat, and, these kinds of things. And that's not going to work for the whole world. This is not going to work as a solution to bigger problems. And so right at the moment I was thinking about these things, Barbara Perea sent me this wonderful quote from one of my favorite authors, Octavia Butler, in a very real sense, and she's talking about one of her characters in one of her novels. In a very real sense, she was an experimental animal, not a pet. What could Nikinj, I don't even know if that's the right pronunciation, do for an experimental animal? Protest tearfully when she was sacrificed at the end of the experiment? But no, it was not that kind of an experiment. So this is something I've been thinking about quite a bit because I really think that we're all experimental animals at this point. And this whole thing that we've been doing with the earth, that the Anthropocene that we're talking about and the kind of problems we have is this giant experiment that we're working on. And I really call on everyone to think about much more radical ideas to move forward. I mean, we're all doing that already, I know, but I'm just reiterating what you're already thinking. And I have a residency coming up next year at the Science University in Philadelphia that's in collaboration with a biotech company, Integral Molecular, and I'm really lucky to be doing that. They work with viruses and bacteriophages and these kind of strange things. So I have one project I'm hoping to pitch. I pitched it to them, but I'm not sure they're buying it, so I'll see if you can help me articulate it a little better. And that's this idea about becoming vulture, which I'm really obsessed with now. So it's a little dark. I hope you can deal with that. But this idea came to me because of I had been doing research on synthetic guts and synthetic microbiomes that have been being developed, where people are curating what goes into it, and I imagined this sort of going forward, coupled with that idea of dysbiotic guts. You get dysbiosis by having lots of antibiotics too, not just Crohn's disease or inflammatory bowel diseases. So if you've had a lot of antibiotics in your life, you probably might have a dysbiosis of some kind, or maybe not. It depends on how long you've been on the antibiotics, et cetera. But vultures have a really interesting gut profile because they actually have lots of bacteria on their faces, which is what they use to enter the animals when they're beginning to eat. But their guts only have primarily two bacteria, Clostridium difficile and Fusobacteria, which in large doses to us is totally toxic. So I'm trying to figure out, what does this mean? Is there a way to think about actually shifting, our digestive system to go more in the direction of dysbiosis, to maybe be able to eat food that is offset. We have so much rotting food in our waste systems. This might be a possibility. I'll close just by thinking about that, and also come back to this idea that I was talking about earlier with Oliver Kellhammer of kind of rooter ecologies and what these plants afford us. These plants that can grow out of toxic urban environments where there were industries dumping who knows what into these soils. These plants have been able to bioremediate in some ways and also hyperaccumulate some of these materials that really need to be taken up from these areas. And so we really need to be studying what they're doing also. And so I also really admire this last, this bottom slide of Oliver's. This is a project of his from 2006, where he simply took an area in a parking lot and the weeds that were already growing there and doing this work of breaking down this concrete, et cetera, et cetera. And he just put it on exhibit as one of his pieces in an exhibition. And I love this idea because I think that we need to be looking at these kinds of ecologies, both internally and externally, and thinking through what we can do for our own futures. So thank you very much.
Well, thank you, Kathy. That's, quite a journey. I think you took us through, and you took yourself during your life as an artist. The first thing I was wondering, it seems like you're quite knowledgeable about all these biological details. How have you been training yourself as being an artist into the details of, well, it's a quite specific trait you are working with?
I'm definitely not a scientist at all, and I have just, and I actually was kicked out of my biology class when I was in high school because I argued with my teacher about the sentience of dolphins. So I have been never had any science background whatsoever, but I became really fascinated in what was happening, partly as a kind of critique of biotechnologies, and then partly to investigate things out of just sheer curiosity. And so I think I came at it from a medical perspective because I've had to learn to treat myself for a very long time. And, that's helped me be able to start studying stuff. So I've embedded myself in laboratories in the last, I don't know how long, 10 years. And so working with scientists and just asking them to teach me. And, we can all learn these things. And I think that it's important now. We have in the States, and I don't know if it's quite the same here, but we have media, arts, and science are completely under attack. I mean, completely under attack by our government to the point where I don't know where this will all go. And I feel like everyone, because of the kind of ways that biotechnology is moving very quickly with advancements, and the ethical questions that raises need to be looked at, we need to think about tooling ourselves with a kind of science literacy. So I've just stepped up to it, I guess.
Something like we need to literate ourselves with media, digital media, but also with science, to be able to ask questions as well, and not only be the ones to listen to the answers other people are coming up with.
Yeah.
And how do they think about your practices, the scientists? Do they learn from you as well? Are you welcome? Do you feel welcome to ask questions? Yeah,
that's a great question. It totally varies, depending upon the individuals. Will DePaulo, for example, completely understands from our working together how important it is to work with artists, not just to be art science communicators, because that's not what this is, but to actually extend the kind of questions that you can ask in a laboratory beyond what science can do, because science has to be very specific by mandate for funding, for everything. And so as an artist, I can come in and start asking these really kind of big, crazy questions, and everybody gets to talk about it. And so it's almost like having a philosopher in the lab, is the way I look at it. And he actually has started artist in residence program as a result of our working together, which is really beautiful. And other people have responded the same. I think it depends on the individual. Scientists are very different. Some scientists are really going to welcome it, and some aren't. And I think everybody should embed themselves in a lab.
In some time of your life. So
Yeah.
let's think about it, which lab that could be. One last question. The working with the red labs, I always feel horrible when looking at, well, those kind of animals, and that we need them so much to, well, in your case, to make your life possible, etc. How did it make you feel as an artist? Did you feel tempted to criticize it more, or were you more like, okay, we just have to do this because it's who we are?
Yeah, your point is really spot on and well taken, because if I could have my way, we would have no animal research whatsoever. But that future is still a little far in our future. I mean, it's not happening right away. And there's lots of movements to move towards that. And I've sat on animal care committees in my universities and stuff to try and change those things. So this is a desperate move to try and figure out how to help those animals that are still stuck in this cycle of being, sorry, that's my timer. See, I was ahead of time. I was really good. Anyway, but the thing is, these systems aren't going to end right away, as I said, and so we have to think about ways to enrich these animals' lives, and so, in a way, I'm hoping that the work itself begins to bring a kind of subtle critique and a way that scientists can start seeing that they're not doing enough, that there are problems for these animals in the lab that extend way even beyond their experiments.
But will we ever be able to do without?
I keep arguing yes, they keep arguing no. No, it's not everybody, but there are problems. Like, one of the big arguments I get is, do you want people to be practicing surgery on humans rather than animals? And I'm like, yes, but they're like, maybe not. So it's a very complicated conundrum. And yeah, when I first started working with the lab rats, I was devastated and super depressed and felt I should be much more of an activist. But I've watched activists work in this area, and their work just gets shut down immediately. They get ignored because of it, which is sad, but true. So
this is more maybe a way to address it better, or at least more than just being activist.
Well, when I first started this work, I had no idea of the life of a lab rat. So, if I can put an exhibit on display for 10 months that has 10,000 people a month coming through it, that shows people what a lab rat looks like and what their conditions are, even though they're in a different environment, there's something to be said. People now know, oh, that's it. We have millions of, sorry, I'm going to go off on a little bit because I'm passionate about this, but there are millions of animals that are called for science, and they're uncounted and they're invisible. So my work, I feel, is to try and bring some visibility to their status and to the work that they do. I don't think I can stop the system. I have tried to work inside and outside the system. And, I would welcome any other strategies anybody has to continue to do some kind of, way of changing it. Because I think you're right. It's a big, hard question.
Well, thank you very much.
Thank you.
We are going to take a break now with something very special. And it's also very non-human. We will having drinks from the non-human bar. Fermented pineapple, tepache, kefir, rose, and cardamom. And this will be brought here on stage. And we can have a drink and we can have a chat for about 20 minutes. And then the program will continue.
Thank you. Yeah.